The overall goal of this cDNA library preparation is to enable successful sequencing of small RNAs that have been stored in formalin-fixed paraffin-embedded tissues.
This method is very useful for analyzing formalin-fixed paraffin-embedded specimens. You extract RNA from up to 18 specimens. You analyze the content of all of these and obtain a profile of all the microRNAs for all these specimens.
To begin the protocol, add 8.5 microliters of previously prepared ligation master mix to each of the 18 individually aliquoted FFPE RNA samples, gently flick the tubes, and centrifuge for 2 seconds. After centrifuging, store the tubes on ice. Use a 3 microliter pipette to transfer 1 microliter of each adapter to the corresponding FFPE RNA samples containing RNA and ligation master mix. Do not pipette up and down, and change tips between FFPE RNA samples. Close each tube, flick them to mix, and centrifuge them for 2 seconds. Set the tubes on ice. Denature the reactions by placing the tubes on a heat block at 90 degrees Celsius for 1 minute and then immediately place them on ice. Next, use a 3 microliter pipette to transfer 1 microliter of the diluted ligation enzyme into each of the 18 RNA samples. Set the ligations on ice and place them in a 4 degrees Celsius cold room for an overnight incubation of 18 hours.